Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Pleasure and an honor again to stand here before you to present a few slides on biosimilars. I would like to thank colleagues in Sandoz for this kind invitation and sponsoring me to present this satellite symposium. So over the next 15 to 20 minutes at maximum, I'll be presenting to you the following objectives. So, history of biosimilars. Then I will be explaining to you the concept of interchangeability in biosimilars. And then I'll be walking you through the translation of the totality of evidence into clinical practice, concluding with some real world evidence, as well as our clinic cases supported by the clinical trials that we have in this manner. So starting with the first part of this presentation, history of biosimilars. So what is basically a biosimilar? So both the European medicine agencies as well as the FDA stated a very clear definition of a biosimilar, in which they mentioned that it's a product which is similar to a biological medicine product from all perspectives. Analytical perspective, pharmacokinetics perspective, pharmacodynamics perspective, as well as clinical characteristics and efficacy and safety as well. So the biosimilar now is implemented actually in the guidelines and the recommendations for the management of several rheumatic diseases. And to summarize both definitions by the European Medicine Agency as well as the FDA, so it says that biosimilars are those products that are highly similar to the reference biologic product based on the submission of quality, safety, and efficacy data. So in another word, biosimilars are another version of the biomolecule of the original medicine. If we go back into history, so the invention and the introduction of biosimilar started almost like 20 years ago, where the European countries initiated a draft that is responsible for the general guidelines that were adopted in the field of biosimilar. Then as you can see here, that in 2006 Sandoz actually launched like the first biosimilar that was approved in the European countries, and then this was followed by other biosimilars and indications. And then in 2010 United States of America followed the same path that was followed in Europe. And luckily in Saudi Arabia in 2015 we had again the first biosimilars that were approved and launched in the country. Following that, we had the first guidelines on the biosimilar that was produced and prepared by the Saudi Food and Drug Authority. And then following that, there was also a Saudi Society for Rheumatology initiative assessing the perception of rheumatologists with regards to the use of biosimilars in Saudi Arabia. So this is the guideline on biosimilar products that was prepared and issued by the Saudi Food and Drug Authority. And this is a study that was conducted by our colleague, Professor Muhammad Umair, in which he assessed the perception of biosimilar biologics and non-medicinal prescription switching among rheumatologists. This was like an initiative by the Saudi Society for Rheumatology. What was noted in this study that were like up to 60 to 70% of practicing rheumatologists, they had actually a good perception with regards to the biosimilars, and they were willing to switch their patients from like an originator medication to a biosimilar medication whenever there is like a possibility of that, or if there is a demand by the authorizing authorities. Now moving on to the second part of this presentation, which is with regards to the interchangeability in biosimilars. So what is meant by interchangeability? So basically interchangeability is either switching or substitution. So for example, if you have a patient who is on an originator A, for example, so switching that patient to biosimilar A, so this is called switching. Or for example, you're simply, but this is because of like a reason, like this is because of like a medical reason. Substitution is basically that you are changing the medication, the originator, to a biosimilar because of the either the practice of dispensing one medicine in place of another without consulting the doctor. So to give you an example of, I think we have all seen that, especially in the Ministry of Health, hospitals, that for example, our patients were on adalimumab reference medicine, and then all of a sudden you notice that patients were receiving like a different medication or a different pen or a different preparation. Basically, it was the same medicine, so it was, without consulting the physician. On the other hand, substituting a patient, from one medicine to another. So this is, for example, if your patient is on an anti-TNF-alpha inhibitor, such as, for example, golimumab, and then all of a sudden this medication goes out of the market because the Ministry of Health decided to stop having like this medicine into the shelves of the Ministry of Health. So then switching this patient from an originator medicine to a biosimilar, this is called like switching. So both are are things that are under the terminology interchangeability. Now, again, the European medicine agencies, incorporated and adopted this concept, and they clearly state, if you look at the two sentences that are highlighted in yellow at the bottom of this slide, that once a biosimilar is approved in the European countries, then it's interchangeable. So this is also applied now in our country through the different healthcare sectors that we have in the country. Now, the third part of the presentation is concerned with regards to the translation of totality of evidence into clinical practice. So what is meant by totality of evidence? So totality of evidence means that utilization of all the resources that are available with regards to the analytical, non-clinical, clinical evidence that can tell you that this medicine is or biosimilar is as good as the originator. And this should be like a stepwise, approach that will convince then the authorities to be, comfortable in prescribing the biosimilar medication. So if you look into the sentence, so again, totality of evidence, as I just mentioned, it's a stepwise, approach that uses the analytical, clinical similarity between the originator and the biosimilar in an attempt to assess the similarity and for possible like interchangeability as well. Now, what happens sometimes that we, when we prescribe a biosimilar to our patient who was, receiving an originator, and then patient goes home with this biosimilar, and then all of a sudden he, finds like a different, package, a different pen, a different color, then patient will be having, some sort of like psychological perception that this medicine might not be like the same, or it might not be working. So it's basically the opposite of placebo, where our patients have negative, expectations on a medication that has not yet been, utilized or used by them, which is called a nocebo effect. And it's very important to address this issue with our patients, because this really decreases the rate of rejection, the possible, nocebo-related anticipated side effects in our patients. Now, this is probably the same that I was just talking about. So let me walk you through a few clinical cases from the practice at Noor Specialist Hospital in Mecca. So this is the first case, Abir, who is a 32-year-old woman from Saudi Arabia. She works as an assistant. Her history started in October 2019 when she presented with seropositive rheumatoid arthritis, who was initially managed with methotrexate that was started at a dose of 10 milligram and then maximized to 20 milligram over like 2-month period, as well as folic acid. 3 months into her treatment, she was still showing active disease, 1 year into her treatment, because this is where like we had the COVID-19, so we were not able to follow up with our patients. She was continuing to see active disease. So at that time, adalimumab originator was added into her treatment regimen. Then from April 2021 to January 2022, so almost like 15 months, we continued to see this patient like on regular follow-up appointments, where she was like continuing to show us a status of the disease that was consistent with remission, and she was on adalimumab reference as well as methotrexate and folic acid. This changed into June 2022 when she presented again for like a routine follow-up, and she would tell us that she received a different pen, adalimumab. It's not the same. That was, Hyrimoz, because in Ministry of Health we had adalimumab Hyrimoz biosimilar that was introduced into the Ministry of Health pharmacies in January 2022. She was using it for over like 3 months before she presents to this follow-up, and she was noticing that nothing has changed in her disease, so she was continuing to be in remission. So we just reassured her. We conducted like a very quick education and counseling that made her like feel comfortable to continue using this medicine. The last time she was seen was in November 2022, where again she was continuing to be in remission, and that was really reassuring. The second case, again from the practice, it's for a patient who is a 55-year-old woman named Amina. She's from Yemen and she works as a housewife. Her history started in December 2022, again at 21, where she again was diagnosed with seropositive rheumatoid arthritis with high inflammatory markers, as well as high disease activity. Again, we started her on methotrexate, folic acid, and prednisone with a quick taper. Almost like 2 months into treatment, she showed mild improvement with mild improvement in her DAS28 score as well. We increased the dose of methotrexate to 15 milligram, and we continued the rest of her treatment. After 3 months, she presented again and was showing clinical as well as radiological evidence of an active disease. And at that time, adalimumab Hyrimoz was added into her treatment regimen. She presented for follow-up in June 2022, where she was showing significant improvement in her clinical as well as radiological aspects of her disease, and we decided to continue her on adalimumab Hyrimoz. The last time she was seen again a few months ago, where she was showing a disease status that was consistent with remission. So this clinical practice is basically based on an evidence that comes from this randomized clinical trial. It is named ADMIRA, which was a multi-center randomized double-blind comparator controlled confirmatory study in patients with moderate to severe rheumatoid arthritis. So patients in this study were randomized either to receive adalimumab Hyrimoz or reference adalimumab for our total period of 12 weeks, which was like the first phase of the study. And then patients in the adalimumab Hyrimoz were continued to be on adalimumab Hyrimoz. However, patients in the reference adalimumab, which are represented here in blue, were switched to adalimumab Hyrimoz after the conclusion of the first week or the first phase of the study. The study was continued for a total of 48 weeks. Now, the primary endpoint of this study was to assess the mean change from baseline in DAS28 CRP scores over 12 weeks. And the secondary endpoint, as you can see here on the right side of the slide, was to continue to notice this change in the DAS28 CRP over a 48 weeks period, which was the second period of the study. Now, as you can see here, that there was clearly no significant difference between the changes in the DAS28 CRP scores in patients on adalimumab Hyrimoz, which is represented here in orange bar charts or bar graph compared to the reference adalimumab, which is presented here in blue. The same thing, was noted with respect to the side effect profile in this patient, where you see that there were no statistically significant differences in the adverse events between both groups. With regards to the immunogenicity, which is the ability of a biological medication to activate the immune system to the extent that it is able to produce anti-drug antibodies as well as neutralizing antibodies, you can notably notice here that the immunogenicity profile, was matching, and there were no differences between adalimumab Hyrimoz compared to adalimumab Humira or reference adalimumab in this study. Again, quality of life was almost different, so there were no changes with respect to facet, or fatigue, or the health, related quality of life, and overall adverse treatment-related, adverse events were like almost similar in both study groups. Moving on to the last, few slides of this presentation, which is, concerned about the real world evidence that we have in this field. So this real world evidence actually comes all the way from, the north, from Denmark, where they had a nationwide, observational study called DanBio. This study studied the effectiveness and safety of adalimumab, biosimilars in patients with, several autoimmune inflammatory rheumatic diseases. So what is happening, or what has happened in Denmark, it also is happening now in Saudi Arabia, especially across like the Ministry of Health hospitals. So there was like a mandatory switch of all patients from originator to biosimilar for cost saving, and this is what's really happening in Ministry of Health in Saudi Arabia as well. This is a very massive registry where they have like more than 50,000 patients. Approximately 1,500 patients were eligible to be enrolled in this study. Out of them, like 1,300 patients were switched to adalimumab biosimilar, and 623 patients were on Sandoz adalimumab biosimilar, because we know that we have other like adalimumab biosimilars other than Sandoz. So the primary endpoint was the rate of retention in these patients. So basically what is meant by the rate of retention is that, so for example, after 1 year, how many of those patients who were on adalimumab biosimilars were continued to be on adalimumab biosimilar and did not require either escalation of treatment or changing of the treatment to another class of action. And there were also some secondary endpoints. So as you can see here, that approximately 90% of patients after 1 year, they were maintained on the adalimumab biosimilars that were started in these patients after like the switch. And 92% of Sandoz adalimumab biosimilar patients were maintained on the same treatment. Now, if you look at the disease remission at 6 months across like the several rheumatic diseases that these patients were treated for. So at the left side of the slide, you see rheumatoid arthritis, you see psoriatic arthritis patients, and you see also patients with ankylosing spondylitis. And if you look at this, forest plot and the line of no effect, you see that all the lines are moving towards the right side, with results that are represented in blue, which indicates that disease was maintained to be in remission in these patients in significant percentages. And as an overall, you see that up to 56% of patients were exhibiting remission rates that was measured by DAS28 as well as ASDAS in patients with ankylosing or axial spondyloarthritis. And again, there was no changes in the disease activity from switch to 6 month post-switch for patients who were maintained on Sandoz adalimumab. And as you can see here, that the disease activity measures that are here in the left side of the slide, you don't see changes in patients across like different disease activity measures in the different diseases such as rheumatoid arthritis, psoriatic arthritis, as well as axial spondyloarthritis. So ladies and gentlemen, if you allow me to summarize the slides that I presented. So I will say that biosimilars allow major cost savings and may increase patient access to biologics. Prospective data in RA and psoriatic arthritis demonstrate high comparability between biosimilar and originator anti-TNFs in terms of efficacy and safety. Rates of immunogenicity with biosimilar and originator anti-TNF alpha inhibitors are similar, and real-world evidence supports the efficacy and safety of switching from an originator anti-TNF to a biosimilar anti-TNF. And I would say that in a neurospecialist hospital, we have approximately more than 100 patients who were either switched from an anti-TNF alpha inhibitor to Sandoz adalimumab, or they were, started on Sandoz adalimumab as an initial treatment. And we continue to see, reassuring and promising, disease statuses in these patients. And I would like to thank you for your kind attention and listening.